These are images of infections caused by bacteria that are resistant to more than one antibiotic. Bacteria that are not killed by multiple drugs are a big problem because they will soon become untreatable. When a bacteria cannot be killed by a drug, it is considered resistant. Bacteria can be resistant to antibiotics intended to kill them. One bacteria that is resistant to multiple antibiotics is the so-called Iraqibacter that is infecting troops in Iraq and Afghanistan. The infections can be acquired in the hospital and are difficult to treat since the normal antibiotic treatments are ineffective. One of the ways bacteria become resistant to antibiotics is by multiplying specific DNA segments that help them fight off these antibiotics. This process is called gene amplification. Gene amplification of specific pieces of DNA affects all organisms. It can even cause cancer in humans. In bacteria, gene amplification increases the ability to infect humans in addition to causing antibiotic resistance. This is one of the reasons why understanding gene amplification is important. Gene amplification can be studied much more easily in bacteria than it can in more complex organisms like corn or humans. But the information we discover from studying how bacteria gain the extra DNA can be applied to these more complex organisms. This diagram shows a bacterial cell. The circle inside the cell represents all of its DNA, and the blue box sitting on the circle represents a specific gene. On the left side, there is a single copy of the gene in the DNA. However, in the right, there are 4 copies of that same gene. How does the cell go from a single copy of this gene to multiple copies? What other parts of the cell are helping with the multiplication of these genes? If we can find this out, we can better understand how to combat the increase in resistance to multiple antibiotics in bacteria. Gene amplification is a very common phenomenon. It occurs in 1 out of every 1,000 cells. On this single plate of bacteria, there are approximately 1 trillion individual bacterial cells, which is about 150 times the population of the whole Earth. 1 billion of these cells on this plate have genes that are found in multiple copy on their DNA. That is equivalent to the entire population of China. The bacterium that we use in the NIDL lab at the University of Georgia to study gene amplification is Acinetobacter bailii, a harmless soil bacterium. This bacterium is a good model system because it is easy to work with and is also closely related to the multiple antibiotic-resistant Iraqibacter mentioned earlier. This soil bacterium can increase its total amount of DNA by about 1/3, from approximately 3 million bases to about 4 million bases. Looking at these bases of the extra DNA, we can determine what other factors within the bacterium can help to increase the levels of the DNA. So far, we have found that different types of DNA recombination are involved in multiplying these genes. As we learn more about how gene amplification happens, we can help to prevent the rise of bacterial strains that are resistant to multiple antibiotics. It is estimated that worldwide, 25,000 people die each year from infections caused by bacteria resistant to multiple antibiotics. With our bacterial system, we hope to better understand how gene amplification affects all organisms and help to decrease the number of deaths due to antibiotic resistance.